Funding for Current Conversations is provided by the University of Oklahoma President's Office, OU Outreach, and World Literature Today. Welcome to Current Conversations. I'm R.C. Davis-Undiano. My guests today for this encore show are Dr. Hong Liu, Dr. Ann West, and Dr. David Schmidtke, two engineers and a chemist trying to unlock the body's secrets. From innovative therapies to new imaging techniques, they are trying to solve the mysteries of good health. Join us for the latest in medical technology next on Current Conversations. You all are scientists doing work, cutting-edge work in your fields. I'm just wondering, at what point in your life and education did you get the bug to do this? You know, maybe discover your passion. And
RC, I think this happened pretty early on for me. I always had this innate curiosity about how things worked in general, and particularly about biological phenomena. I remember tinkering around with chemistry sets when I was young. I received a microscope for my 13th birthday, and I had an aptitude for science and math, but I think it really wasn't until college-level classes that I realized I could actually make a career out of this.
So there wasn't a single day where something dramatic happened. It was just a gradual realization.
That's correct.
Yeah.
Yeah.
Yeah, well, I'm an engineer. Actually, ever since a little kid, I'm a hands-on type of person. I'm interested in building something up and observing how something works. Before college, I worked a few years as a lathe operator in a manufacturing factory. And therefore, I select majoring in engineering as my college education. Mm-hmm. And as well, I'm interested in observing how physicians save people's life. Mm-hmm. And I pay high respect to their dedication and their tremendous knowledge and their work. And it seems to me, while I'm growing in my career, that joining engineering and biomedical sciences, biomedical engineering is a wonderful career path.
Okay, Dave.
For me, it was as an undergrad. So I was good, in high school, math and chemistry. And my original plan was to go to medical school. Mm-hmm. So I was on the path there. And then my junior year, it was right before the summer, I saw a flyer for doing undergraduate research in a bioengineering lab. And so I went and got the position and started doing this bioengineering research and realized I could still have an impact and be in the medical field without having to be on call 24 hours a day like a physician. And so that sort of led me down the pathway of bioengineer.
I was talking to a graduate student, PhD student in biomedical research recently. And I said, another student came in on the conversation and said, what's the difference between what you do and what med students do? And rather proudly, the PhD student said, medical students do a lot of memorizing. We understand what's going on. So could you talk about your work a little bit? Could we start with you? And what kinds of questions are you asking in your work? Yeah.
My research group is interested in general how cells have the ability to sense their environment, their surroundings, and particularly at a molecular level. So we're interested in how proteins that reside at the surface of the cell serve almost like a receptor molecule and can perceive outside stimuli and then transmit that information into the interior of the cell, all the way down to the nucleus and the DNA, and reprogram gene expression so that cells can adapt to whatever the change in the environment is. And so at a molecular level, this is quite intricate. It's a pathway, and proteins have to communicate with each other. And so we're interested in what the proteins look like, what their shapes are, because this tells us something about how they function. We're interested in how they recognize each other and not other proteins in the cell. And what we study mostly are in bacteria and fungi. if we understand how these pathways work normally, we can imagine in the future designing drugs, for example, that would be antibacterial drugs, antifungal drugs that could help humankind.
I see. So if these
Yes.
are bad guy cells, like
Yes,
bacteria,
that's right.
and
Pathogens.
you could do something so that they sense their environment differently,
Right.
and maybe they sense a hostile environment, or something to help to remove them from that place. That could be a
Exactly.
very helpful thing to do. How do you study proteins? Aren't they? Those are very, very small, right?
That's right. We cannot see what a protein looks like by our naked eyes. We cannot even use a light microscope. They're even smaller than that. So the resolution that we need to look at it requires X-ray radiation. And if we can crystallize pure proteins and shine X-rays through these crystals, through a series of computational methods, we can deduce their three-dimensional structure. So it's really quite revealing when you're able to see and imagine what your favorite molecule looks like. It tells us a lot about how they function.
You know, when you say we don't know what they look like, as a non-scientist, I'm sort of starting to understand these days that visual understanding of things is becoming old-fashioned. Do you mean literally we want to get a sense of an actual picture, a 3D picture?
Imagine if you had a bicycle sitting here in your studio and we didn't know what that looked like. Could you imagine trying to figure out how it worked without knowing that there were two round wheels and pedals that make the wheels go and handlebars that steer the bicycle? It's sort of
there's an operation
analogy, right?
In the protein. Seems like so many things are coming back to protein. We'll talk about that in a moment. So, what's the center called that you direct?
I'm a director of the Oklahoma Center of Biomedical Research Excellence. It's COBRE for short, in structural biology. So the field that I'm in is a general term, structural biology.
How does that connect with your work? I mean, are you partnering with people in that?
Yes, yes. There are many of us that have a common interest in learning what the three-dimensional structures are of the proteins that we're studying. And the proteins can be involved in many, many different cellular processes, but we have a common link there in terms of using X-ray diffraction methods and wanting to know what the molecule looks like and how that dictates how they work.
Okay. Hong, could you tell us about your work?
Well, I'm working in cancer imaging. I'm a biomedical engineer and focus our research in the imaging technology development. Give an example, for instance, breast cancer is one of the leading cancer deaths right here in the United States, but also worldwide.
240,000 a year, I think, just in the U.S.
Oh, yeah, sure. But breast cancer screening and early diagnosis has played an important role, as well as better treatments. And as we can see in the past 10, 15 years, ever since 1990s, the death rate has been continuously dropping. And technology played a role. Introduction of the digital mammography, for instance, in the end of the 1990s, beginning of the 2000s, as well as other new technology, has been helping the physician to better diagnose the type of the cancer, but more importantly, through screening program to find those suspicious tumors. So early detection actually is the key. That is the area we work very closely with the physicians, trying to improve the accuracy, specificity, sensitivity of the mammography screening and the diagnosis.
I get the impression that in your research, there's always a kind of a tension between wanting to get more information with a more intense look, more information coming back, and then the dangers of increased X-ray that could potentially do some harm. It seems like there's always that tension between trying to get more information, but then trying to reduce the X-rays without...
Oh, yeah, sure. Mammography is based on X-ray. It's still, nowadays, considered one of the important or effective tool, particularly for screening. And of course, X-ray has certain circumstances may have some harm to the patient. But like all engineers as well as physicians, and we all understand that there is always a risk and a benefit factors. And we try not only our own group, but as well as the engineers, as well as physicians nationwide, making great scientific scientific effort to reduce the patient radiation, as well as to improve, at the same time, to improve image quality.
I guess one door to getting more information or reducing the radiation would be to introduce nanoparticles that could be located in the areas of tumors, and then they can, the refracted light can give different kinds of phase information.
Well, a variety of the techniques that has been explored and utilizing a nanoparticle-based contrast agent with the different modalities, not only with X-ray, but also with such as MRI, has been explored. So people are making great effort, seeking new technology to improve the early detection of the cancer. Early detection play an important role in the cancer diagnosis.
Okay. Dave, could you tell us about your work?
So we have research going in a couple different areas, and I might categorize it generally as biomedical technologies. So for instance, one focus of my lab is in the area of cell adhesion, particularly in the area of cardiovascular system. And so what we're trying to understand is how do cells such as platelets or white blood cells, how do they adhere to, natural surfaces such as your blood vessels if you get a cut or inflammation, but also just as important to artificial substrates. So if you put a new medical implant, in some cases you want cells to adhere, so you might stop the bleeding. In other cases, you don't want them to adhere, so you don't get a clot in your cardiovascular system. And then the other primary research area that we do work in is in the area of biosensors. And probably the best example is developing a glucose sensor for diabetics. And for diabetes, the main goal is to control your sugar levels as close to normal levels as you can, because there's numerous studies that have shown if you can control those near normal, you prevent a lot of the secondary complications, such as amputations or neuropathy or kidney disease. And so we are developing a couple different detection systems that can measure glucose levels continuously.
I have a question for you. I think I understand about the dangers of cell adhesion. Yeah. Certain kinds of cells adhere to blood vessel linings and make a new lining that could result in hypertension. There could be blood clots that could come out of that, et cetera. When you talk about nanotechnology and sensing, is the idea to get an as early as possible sensing of something? Like, why wouldn't I just wait for the doctor to do a blood test and find out what my dangerous levels are?
Well, there are a couple of reasons. One is, particularly if you're talking about a stroke, you don't want to wait until you get to the hospital or see a doctor. You'd like to know right away if they could develop something like that. Where nanotechnology, I think, would become important as far as how these particles interact with cells is this, quote, silver bullet, the silver bullet for cancer. One of the problems of current therapies, is chemotherapy, you give the drug and it goes everywhere. It affects both the cancer cells and the normal cells. And if you could understand the biology of how to target only the cancer cells, you could deliver a high dose of the drug to kill only the cancer cells and not have all these other side effects.
Like a nanoparticle would carry something to them and then embed, or?
Yes, so there's a number of researchers that are developing that, some on the Norman campus in the Biomedical Engineering Center, also at the Health Science Center, and they're looking at nanoparticles that are coated with specific molecules that will direct them to certain cell types, but these particles also carry drugs that they can deliver locally.
Oh, that's exciting. Could you all tell me, what do you think is where are things likely to go in your field over the next 10 years? And what breakthroughs, people are going to hear it here today, what breakthroughs did you predict that we're going to see maybe in another couple of years in your field?
Technology has really been improving and driving our field, and to a point where we can handle larger scale projects. For example, we refer to the Omics revolution, where the Human Genome Sequencing Project has allowed us to now look at what those genes encode. Those genes encode RNA and then proteins, and so the proteins are the workhorse of the cell. Researchers in my field are tackling this at a total cellular level and trying to understand the structures of all the proteins in a cell. And this will provide information to the whole research community rather than trying to tackle one at a time, for example.
Okay.
Yeah, we are continually working, improving the accuracy of the imaging devices. And as well, we are working together with people in different fields, developing different biomarkers. And joining the forces of imaging technology with biomarkers, we could able to identify the early sign of the cancer. Like I mentioned before, that early detection is the key. Many cancers are curable disease as long as we can find them at
What would
early
be
stage.
an example of a biomarker? Can you just give us an example?
Well, biomarkers can help identify where the tumor is, and particularly when they growing in the very beginning, the tab. And for instance, we are working in our laboratory and some other laboratories based on a nanoparticle technology. And those functional coatings, they are designed to attach with some molecules and even at the protein level. Mm-hmm. And those nanoparticles will carry together with the functional coating to those tumor areas. And we are utilizing our technology, either X-ray or optics, to see where are they and how much are they. That would give us a sign where the tumor is, at which stage they are growing.
Smart particles that will seek out the disease. That is
wonderful
really
summarization.
Yeah.
Mm-hmm.
So I think there's two main things, and they're somewhat related. Is that one? I think the approach that one therapy fits all is going to go away. People are realizing that when you say breast cancer, there's multiple forms of breast cancer, and so I think it's really going to move towards personalized medicine, that we've got this person has a unique signature of cancer versus this other person, and we're going to have to treat them maybe a little bit different than the other person.
You mean like a genetic match?
Right, but maybe some genetics, maybe some of these biomarkers. We're going to look at different protein levels in the blood and see this person has elevated A protein, this is elevated B protein. The other thing that I think we'll see, and I think you've already seen it in the diabetes field, is point-of-care testing, that now people at their home can test things. If you look at the evolution, back in the 70s, it was urine test strips where you had to see if you had protein in your urine. And now you have not only the finger prick assays, but there are continuous monitors that can give you a continuous readout of your sugar levels that you're getting too high or too low. And so those same type of technologies could be applied to cardiovascular disease, to cancer, and I think really that's where it's moving.
You all are part of research teams. That's just your life. You do everything in conjunction with them. Is that part of what's speeded up the process of discovery?
Sure. Collaboration is a key. And we are working in the field, in the area, or in the time that we have to work together. In our own field, we work very closely with the physicians. I always tell my students that in our laboratory, as engineers, we can always build something. But if we are working together with the physicians, we can build something that have a real clinical application. And we will always have patients in mind. And as engineers, we work together with physicians on every steps.
Our research has become very much more interdisciplinary. It's no longer the case that you could be narrowly focused, let's say, or trained as perhaps just a chemist or just a biologist. The work we're involved in now has a history in physics, a foundation in chemistry, but the applications are in biology. And so we collaborate because we either need people with complementary expertise, or we want to share expertise in a synergistic way and be able to use resources and get the types of sophisticated instruments we need So there is a great sense of cooperation for the greater good.
It must affect the way you think when you're working in a team, I would think. When I was an undergraduate, I remember there was a lot of talk about interdisciplinary work, but as far as I could tell, it was sloganeering. There really wasn't much of anything going on. You all don't do much of anything except in collaboration with somebody else. You must have to think differently when you're crossing those interdisciplinary lines and depending on other people to support your work, I would think. A different kind of thinking.
It's very refreshing talking to other scientists who are outside of your field and trying to understand what they know and vice versa.
Yeah, in my understanding that first we have to be good in our own profession. And then we have to learn the other field's language. In our case, we have to learn how to listen to the physicians and how to listen to the biologists, the molecular biologists.
Is there, Dave, do you think is there a profile of an effective scientist, given the environment, working in collaboration, the kind of interdisciplinary research these days? In your mind, is there a profile of a kind of person that's, in terms of their skill sets, personal tendencies, kind of fits this?
Oh, is there a single one? You know, I'd say there are a lot of, if you look at scientists, there are a lot of pathways to success, you know. But I would say, you know, one characteristic that you see with probably some of the most successful scientists is typically they focus on one thing and they do it well and they try to be the best at that. And part of that being the best is talking to other people in other fields and saying, well, I'm an expertise in here, but I need a little bit more cell biology background, so I'll talk to Ann West. Or I need more of an imaging background, I'll talk to Hong Lu. But I think, you know, having that drive to accomplish something.
It's what Hong said. You go very deep in one area and then join the conversation with people in other areas.
Yes, yeah.
Is there anything that, let's say you could go back and tell the young version of yourself, the young scientist just getting started, something that you've learned in your years working in the lab. What would that be? What would you go back and tell the young version of yourself?
Well, I always tell my students that build a solid fundamental background through the formal education. Pay attention to the classes, but as well continuously improving your self-learning skills. Considering many years ago, well, you and we were in colleges. And before I came here, I take a look of one of the things those cards, we punch holes in order to run computers. That is in the end of the 1970s.
It took up whole rooms.
Absolutely, absolutely. But self-learning skills is very important. And as well, I feel that there are always students come to my laboratory and say, hey, Dr. Liu, I'm interested in what you are doing. Can I join your group? I always ask them that think about two things. One thing, is this something of your interest, or do you have an adequate background? Are you strong? Have strength? Do you have a strength in this area? So not only join a career or take a job just because they pay more. You have to be interested into it, and you have an adequate background to do it. And then every morning you'll be very happy to go to your work.
The kind of work that you do, if you don't really fit in the job, where it's pleasurable to do it, and you're going to work long hours and do what it takes, it's just not going to work.
I guess the same thing. Perhaps I never truly learned molecular biology, although I have to work together with molecular biology. I'm happy I was not in that field.
Well, that's the beauty of our career is that we get to be lifelong learners, essentially, through our research, because we can collaborate with others and remain on the cutting edge and learn from other scientists in different fields. But I agree exactly with what Hong Lu has said, you know, a firm foundation at a younger age in terms of formal education, but that opens doors to all these other opportunities, and you got to find your passion, and it could be different for different people.
You can find your passion and hook onto
find
that.
Your passion, that's the key.
That's one of the keys is what is your passion? Because it'll get you through those long days when most experiments don't work. And really the PhD is more the patience than anything else. But your passion, and then what are you good at? You don't want to be trying to force a square peg in a round hole.
It really needs to fit.
You look
for
at
you.
what are your abilities, what are you good at? So, I'd say, what is your passion and what are you good at?
David, is there something, though, that if you go back and talk to the younger version of yourself, you'd say, don't worry about that so much. Now that I'm a scientist, I can see this is what you need to focus on. Go with me on this. Is there something you would go back and tell young Dave?
What I would tell actually is it comes from a book that my wife and my sons have just finished over the summer, and it's a biography on Ben Carson. And he is this
Ben Carson is
the famous neurosurgeon.
Okay.
Separated Siamese twins, and he's got an acronym saying, "Think big." So not only think big, but then the think big. The T stands for talent. We all have God-given talent. Find out what yours is. H, be honest. I is find inspiration from others, insight. The N is be nice to others, don't step on people.
Yeah.
K is knowledge, gain as much knowledge as you can. That's really what's valuable, is the knowledge you gain. B is for books, read as many books as you can. I is in-depth study, not superficial like we just said, go in-depth. And then the G is, thank God for your blessings and use your blessings to serve others.
To be allowed to do this wonderful work.
Amazing you can memorize all of that together.
Well, it hit us. My wife and I love that. We love that. I like that acronym. I think that has a lot of the characteristics that we talked about. Passion, what are your talents? And I think that it's a great model.
I think Dave touched a very important points, that we have to have a big picture in our mind, but also move forward toward that goal step by step.
I think on that note, I want to thank you all. This has been wonderful. I know I'm going to hear a lot more about your work over the years, your wonderful work. Thank you so much for being here. That's all we have time for today. Join us next time for more current conversations. Thank you for being with us.